in this particular gene, but the WHO catalog doesn't record this mutation. So now what? Do we call it resistant? If we look at the catalog, the mutations surrounding it all cause resistance, but this has never been reported in the world before. So I'm just trying to show you the complexity that exists, that in fact, we need everybody in the world to contribute their data to the WHO catalog so that that catalog can be comprehensive. In this instance, we basically know that most mutations in PNCA confer resistance to pyrazinamide. So in an expert panel, we would have said we want to be cautious. We will not use pyrazinamide to treat this patient. Moving slightly forward, but on the same theme, bedaquiline, which is, as I mentioned, this very important new drug. And here is a systematic review done by a postdoc of mine and a PhD student in Antwerp. And this really shows, and I'm not going to go into detail, that there are a lot of mutations in this gene that is associated with the resistance, but not all of these mutations confer resistance. So again, we really need more and more data contributing to be able to come up with a very good catalog that can guide therapy. So that's the big limitation at the moment about drug using next-gen sequencing to guide therapy. I'm going to give you quickly an idea of another study that is just about to start, and it is basically saying, can we use this duplex assay together with next-gen sequencing and do it in a diagnostic and academic environment so that we can move next-gen sequencing closer to the actual diagnosis, it as the routine for the standard of care in the future. This assay, as I mentioned, looks at a defined method, etc. And the assay, basically the information goes up into a cloud and it can give a very nice report that can be sent to the clinician or the clinical care authority who can decide on whether the regimen needs to be changed. It can give us an answer basically in around 17 hours, but we still need some research. We really need to understand whether or not we can use sputum specimens or whether we need to still use cultures. Most importantly, when one's introducing an assay in a diagnostic environment is not to disrupt this diagnosis. So we're trying to be able to run it in parallel rather than right within the diagnostic algorithm in the beginning. And hopefully when we have the information, it will be contributed to the WHO guidelines on using this assay going forward. So the complexities of implementing this assay into routine is, as I've just mentioned, not to interfere with current workflows. How big is the footprint? Do we have a big enough lab? Do we have the right flows? We don't necessarily have skilled staff currently in these labs that are skilled in the bioinformatics and next-gen sequencing. We really need automation rather than having a finicky, difficult assay. How to interpret the data, as I mentioned, the WHO catalog, and how will the doctors react to this new set of information? So this is really the challenges that are lying ahead of us. So the take-home messages, I think, from these last few slides is we are moving rapidly towards next-gen sequencing platforms, but we need to standardize that everybody's doing the same thing rather than each per group is doing something different. We need to have more and more studies that are implementing next-gen sequencing into the standard of care. There needs to be a global effort to update the mutation catalog. As I've mentioned, automation is essential. And we have one big critical barrier, and that is DNA extraction is not efficient from mycobacteria, given its really robust cell wall. I'm going to leave you with one example. Another example, perhaps, of bedaquiline resistance. Here we have a patient who is diagnosed with rifampicin resistance. We're using targeted deep sequencing here. We're looking at the patient. The patient stopped taking therapy for 9 months, came back, and we can see the emergence of this resistance in different clones. The patient took therapy for a while and then stopped, or was well and couldn't produce a sputum, and then relapsed while on therapy. And we can see how during these periods, we are selecting for resistance. We desperately need to find ways to rapidly identify bedaquiline resistance so that we don't lose it as the most important drug in fighting rifampicin resistance in Africa and in the world. At that point, I'm going to stop. I'm done 40 minutes and I'm open for questions. I would just like to acknowledge all my funders that have contributed to this research and all of the colleagues in the world that are partnering with me and collaborating in my research. Thank you very much indeed.
Thank you. Thank you very much, Professor Warren, for that very interesting insights into the mechanisms for drug resistance and the genomic mechanisms. And I think I will just dive straight into the question and answer panel. We've got quite a number of questions, and I will go through them in the order in which they were asked. I think the first question was from from Daria de Villiers, and Daria de Villiers is actually delving into the topic of wastewater surveillance. And the question is, if you have tried to combine the work that is being done by the SA Medical Research Council team that is monitoring COVID-19 variants. So saying they're using wastewater samples to generate bigger sets of data, and reckons that this is a massive opportunity to identify drug-resistant strains and clusters of these strains. So I don't know your comment on that.
I think